Hi all. Presentation for today is going to be on cell. The content of the presentation: structure of the cell, cell membrane, cytoplasm, organelles in the cytoplasm, organelles with limiting membrane, organelles without limiting membrane. Introduction: all living things are composed of cells. A single cell is the smallest unit that has all the characteristics of life. Cell is defined as the structure and the functional unit of a living body. The general characteristic of a cell: each cell in the body needs nutrition and oxygen, produces its own energy necessary for its growth, repair, and other activities, eliminates carbon dioxide and other metabolic waste, maintains the medium, that is, the environment, for its survival. And it shows immediate response to the entry of invaded like bacteria or other toxic substances into the body, reproduces by division. A tissue is defined as the group of cells having similar function. Tissues are classified into four major types, which are called as primary tissues. The primary tissues includes muscle, nerves, epithelial tissue, connective tissue. So these are the four types of tissues: connective tissue, epithelial tissue, muscle tissue, and nervous tissue. The organ. An organ is defined as a structure that is formed by two or more primary types of tissues which exudate at the function of the organ. Some organs are composed of all four types of primary tissues. The organs are of two types, that is tubular or hollow organ. Another one is compact or parenchymal organ. Now, what is a system? The organ system is defined as a group of organs that work together to carry out a specific function of the body. Each system performs a specific function. Going into the structure of the cell. Each cell is formed by a cell body and a membrane covering the cell body called as a cell membrane. Cell body has two parts, namely nucleus and the cytoplasm surrounding the nucleus. Thus, the structure of the cell is studied under three headings. One, the cytoplasm, cell membrane. Another, cytoplasm. The third, the nucleus. Going into the cell membrane. A cell membrane is a protective sheet enveloping the cell body. It is also called as the plasma membrane. This membrane separates the fluid outside the cell called as the extracellular fluid and the fluid inside called as the intracellular fluid. It is a semi-permeable membrane. So, there is free exchange of certain substances between the extracellular fluid and intracellular fluid. Thickness of the cell membrane varies from 75 to 111 micron. Composition of cell membrane. Cell membrane is composed of three types of substances: protein, lipid, and carbohydrates. Protein is composed of 55%, lipid 40%, carbohydrates 5%. Going into the structure of the cell membrane. Cell membrane is called as a unit membrane or a three-layered membrane. Three layers of the cell membrane, namely one central electron-lucent layer and two electron-dense layers. The two electron-dense layers are placed one on either side of the central layer. The central layer is a lipid layer formed by lipid substances. The other two layers are protein layers formed by proteins. Cell membrane contains some carbohydrate molecules also. Lipid layers of the cell membrane. It is a bilayered structure. This is formed by a thin film of lipids. It is fluid in nature. Major lipids are phospholipid and cholesterols. So phospholipid, they are the lipid substances containing phosphorus and fatty acid. Their molecules are arranged in two layers. Head protein is the polar end, which is the hydrophilic, and the inner portion is called as the tail portion, non-polar end called as the hydrophobic. This is a cell membrane structure. Going into cholesterol. Cholesterols are arranged between the phospholipid molecules. Phospholipids are soft and oily structures. Cholesterol helps to pack the phospholipids in the membrane. It is responsible for the structural integrity of the lipid layer of the cell membrane. The function of this lipid layer is: lipid layer of the cell membrane is a semi-permeable membrane and allows only the fat-soluble substances to pass through it. Thus, the fat-soluble substances like oxygen, carbon dioxide, and alcohol can pass through this lipid layer. The water substances such as glucose, urea, and electrolyte cannot pass through this layer. Protein layer of the cell membrane. It is an electron-dense layer. It covers the two surfaces of the central lipid layer. Gives protection to the central lipid layer, mostly the glycoproteins. Protein molecules are classified into two categories: the integral protein and the peripheral protein. The integral protein is otherwise called as the transmembrane protein. The peripheral protein is otherwise called as the peripheral membrane protein. Integral proteins are the proteins that pass through the entire thickness of the cell membrane from one side to the other side. These proteins are tightly bound with the cell membrane. Examples: cell adhesion protein, cell junction protein, some carrier proteins, channel proteins, some hormone proteins, antigens, some enzymes. Peripheral proteins or peripheral membrane proteins are the proteins which are partially embedded in the outer and the inner surface of the cell membranes and do not penetrate the cell membrane. Peripheral proteins are loosely bound with integral proteins or lipid layer of the cell membrane. So, these protein molecules dissociate readily from the cell membrane. Example of peripheral proteins: proteins of cytokine, some protein carriers, some enzymes. The function of the protein in cell membrane: it provides the structural integrity of cell membrane, helps in diffusion of water-soluble substances like glucose and electrolyte, helps in the transport of substance across the cell membrane by means of active or passive transport. It pumps some carrier proteins act as a pump by which ions are transported actively across the cell membrane, serves as a receptor site for hormones and neurotransmitters. For enzymes, some of the protein molecules from the enzymes and control chemical reactions within the cell membrane. Some proteins act as an antigen and include the process of antigen formation. They are responsible for attachment of the cell to the neighboring cell or the basal membrane. Carbohydrates of the cell membrane. Some of the carbohydrate molecules present in the cell membranes are attached to the proteins and forms glycoprotein. This is otherwise called as a proteoglycan. Some carbohydrate molecules are attached to lipid and forms glycolipid. Carbohydrate molecules form a thin and loose covering over the entire surface of the cell membrane called as a glycocalyx. Function of a carbohydrates in the cell membrane. Carbohydrate molecules are negatively charged and do not permit the negatively charged substances to move in and out of the cell. Glycocalyx forms the neighboring cell, helps the tight fixation of the cell with one another. Cytoplasm. It is the jelly-like material formed by 80% of water, contains a clear liquid portion called as a cytosol and various particles of the different shape and size. These particles are protein, carbohydrates, lipid, or electrolyte in nature. Cytoplasm is made up of two zones: the ectoplasm and the endoplasm. The ectoplasm is a peripheral part of the cytoplasm situated just beneath the cell membrane. The endoplasm is the inner part of the cytoplasm interposed between the cytoplasm and the nucleus. The cytoplasm. Organelles in the cytoplasm. They are cell structure embedded in the cytoplasm. Some organelles are bound by limiting membranes and others do not have limiting membranes. Each organelle is having a definite structure and specific function. The organelles with limiting membranes are endoplasmic reticulum, Golgi apparatus, lysosome, peroxisome, centrosome and centrioles, secretory vesicle, mitochondria and nucleus. Organelles without limiting membranes are ribosome and cytokines. Now we will see about the organelles with limiting membrane. First, endoplasmic reticulum. Endoplasmic reticulum. Network of tubular and microsomal vesicular structure which are interconnected with one another. The endoplasmic reticulum forms the link between the nucleus and the cell membrane by connecting the cell membrane with the nuclear membrane. It is covered by a limiting membrane formed by protein and bilayered lipid. The lumen in endoplasmic reticulum contains a fluid medium called as the endoplasmic matrix. Rough endoplasmic reticulum. It is a rough, bumpy or bead-like appearance. Rough appearance is due to the attachment of granular ribosome to its outer surface. Hence, it is called as a granular endoplasmic reticulum. Rough endoplasmic reticulum is vesicular or tubular in structure. The smooth endoplasmic reticulum is with smooth appearance. It is also called as an agranular reticulum. It is formed by many interconnected tubules, so it is also called as a tubular endoplasmic reticulum. The function of the endoplasmic reticulum. The rough endoplasmic reticulum, synthesis of protein, degeneration of onouts, organelles, that is autophagosome. Smooth endoplasmic reticulum, the functions are synthesis of lipids and steroids, role in cellular metabolism, storage of metabolism of calcium, catabolism and detoxification of toxic substances. Going into Golgi apparatus. It is a membrane-bordered organelle involved in the process of protein present in all the cells except red blood cells. Each cell has one Golgi apparatus, consists of 5 to 8 flattened membranous sac called as a cisternae. Situated near the nucleus, it has two ends of the phase, namely the cis phase and the trans phase. The function of the Golgi apparatus, it helps in the processing, packaging, labeling and delivery of protein and lipids. Lysosome. The lysosome is a membrane-bound vesicle organelles found throughout the cytoplasm. The lysosomes are formed by Golgi apparatus. The enzyme synthesized is rough endoplasmic reticulum are processed and packed in the form of small vesicles in the Golgi apparatus. Then these are pinched off the Golgi apparatus and they become the lysosome. Among the organelles of the cytoplasm, the lysosome have the thickest covering membrane. The membrane is formed by a bilayered lipid material. It has many small granules which contains the hydrolytic enzyme. The types of lysosome. There are two types of lysosomes, primary lysosome and secondary lysosomes. Primary lysosome, which is pinched off the Golgi apparatus. It is inactive in spite of having hydrolytic enzyme. The secondary lysosome, which is active lysosome, it is formed by the fusion of primary lysosome with the phagosome or endosome. The functions of lysosomes, they help in degradation of macromolecules, degradation of O-node organelles, removal of excess secretory products, secretion of perforin, granulism, melanin, and serotonin. Peroxisomes. Peroxisomes or microbodies are the membrane-limited vesicle-like lysosomes. Peroxisomes are pinched off from the endoplasmic reticulum. Peroxisomes contain some oxidative enzymes such as catalase, urate oxidase, and D-aminoacidic oxidase. The function of peroxisomes. It helps in breakdown of excess fatty acids. It helps in detoxification of hydrogen peroxide and other metabolic products. Oxygen utilization, acceleration of gluconeogenesis, degradation of purine to uric acid, role in the formation of myelin, role in the formation of bile acids. Centrosome and centrioles. These are membrane-bound cellular organelles situated almost in the center of the cell close to the nucleus. It consists of two cylindrical structures called centrioles, which are made up of proteins. Centrioles are responsible for the movement of chromosome during cell division. The secretory vesicles. These are organelles with limiting membrane and contains the secretory substances. These vesicles are formed in the endoplasmic reticulum and are processed and packed in the Golgi apparatus. Secretory vesicles are present throughout the cytoplasm. When necessary, these vesicles are ruptured and secretory substances are released into the cytoplasm. Going into mitochondria. It is a membrane-bound cytoplasmic organelles concerned with production of energy. It is a rod-shaped or oval-shaped structure with a diameter of 0.5 to 1 micron. It is covered by a bilayered membrane. The outer membrane is smooth and encloses the content of mitochondrion, contains enzymes such as acetyl-CoA synthesis and glycosyrol phosphate acetyl transferase. The inner membrane is folded in the form of shelf-like inward projection called as the cyste, and it is covered the inner matrix space. Cyste contains many enzymes and other protein molecules which are involved in the respiration and synthesis of adenosine triphosphate, which is ATP. Mitochondrion moves freely in the cytoplasm of the cell. It is capable of reproducing itself. Mitochondrion contains its own deoxyribonucleic acid, that is the DNA, which is responsible for many enzymatic actions. In fact, mitochondria is the only organelle other than nucleus which has its own DNA. Functions of mitochondria. It helps in the production of energy, synthesis of ATP, initiation of apoptosis, storage of calcium, and detoxification of ammonia in liver. Going into organelles without limiting membrane. Ribosome. Organelles without limiting membrane. Granules and small dot-like structure. Ribosomes are made up of 35% of protein and 65% of ribonucleic acid. RNA present in the ribosome is called as a ribosomal RNA, which is rRNA. Ribosome are concerned with the protein synthesis in the cell. The types of RNA, there are two types: ribosome that are attached to the rough endoplasmic reticulum and free ribosome that are distributed in the cytoplasm. Going to the functions of ribosome, it is a protein factories because their role is the synthesis of protein. And messenger RNA carries the genetic code for protein synthesis from nucleus to the ribosome. The ribosome in turn arranges the amino acid into small units of protein. Ribosomes attached to the rough endoplasmic reticulum are involved in the synthesis of proteins such as the enzymatic proteins, hormonal proteins, lysosomal proteins, and the proteins of the cell membrane. Free ribosomes are responsible for the synthesis of protein in hemoglobin, peroxisome, and mitochondria. The cytoskeleton structure. The organ present throughout the cytoplasm, it determines the shape of the cell and gives support to the cell. It is a complex network of structure with varying size. In addition to the determining the shape of the cell, it is also essential for the cellular movement and the response of the cell to the external stimuli. Cytoskeleton consists of three components: microtubular, intermediate filament, and microfilament. The microtubular are straight, hollow, and tubular structures of cytoskeleton. These organelles without the limiting membrane are arranged in different bundles. Microtubular are formed by bundles of globular protein called as a tubule. The function of microtubules, it determines the shape of the cell, gives structure strength to the cell, acts like a conveyor belt which allows the movement of granules, vesicle, protein molecules, some organelles like mitochondria to different parts of the cell. Forms the spindle fibers which separates the chromosome during the mitosis. They are responsible for the movement of centrioles and the complex cellular structures like cilia. The intermediate filaments forms a network around the nucleus and extends the periphery of the cell. The intermediate filaments are formed by rope-like polymers which are made up of fibrous protein. The function of intermediate filaments helps to maintain the shape of the cell. These filaments also connect the adjacent cell through desmosome. The microfilaments are long and fine thread-like structures. These filaments are made up of non-tubular contractile protein called as actin and myosin, present throughout the cytoplasm. The function of the microfilaments are give structural strength to the cell, provides resistance to the cell against the pulling force. They are responsible for cellular movements like contraction, gliding, and cytokinase. Thank you.